Good morning, everyone. Welcome to today's Harley Hotchkiss Lecture. As you know, the Harley Hotchkiss Memorial Lecture is organized by the Canadian Centre for Behavioural Neuroscience at the University of Lethbridge since 2013. The series was designed to bring in neuroscientists from all over the world to Southern Alberta to present their research and meet the department. The series honors Harley Hotchkiss, Canadian businessman and philanthropist that supported scientific research. In the 10 years of existence, it has brought in over 100 renowned scientists to Lethbridge, making possible a most valuable exchange of ideas and information. Today, we have with us Dr. Michael Levin from Tufts University, and telling us a little bit about his career and his accomplishments, we have Dr. Arthur Lupczak. Dr. Lupczak?
Yes, so it is a great pleasure to have Dr. Levin here. So he got a bachelor degree in computer science and in biology, and he got a PhD from Harvard University. Among many awards he received, he is distinguished professor in biology at Tufts. He's endowed chair, and he's director of Allen Discovery Center in Tufts and Tufts Center for Regenerative and Developmental Biology. And today he will talk about how intelligence can emerge from cellular swarms. And here I would like to add my personal note that his work really inspired me how I think about brain, but it is just a collection of smart, intelligent neurons interacting with each other to create our behavior. And it is not only me. When I spoke with Brian Kolb, what he would do if he would be just starting his career, he told me about one of the videos of Dr. Levin. So pay attention because this is really exciting work. Welcome very much, and thank you for joining us, Dr. Levin.
Thank you so much. Thank you for that kind introduction, and thank you for the opportunity to discuss these ideas with all of you. Afterwards, if anybody's interested in downloading the primary papers, the data sets, the software, everything is here at this site. So today I would like to transmit the following main points. We will first talk about this idea that intelligence, and in particular navigation of arbitrary problem spaces, applies well beyond conventional brainy animals moving in three-dimensional space. I'm going to stress this idea that we are all collective intelligences and try to stretch some aspects of neuroscience beyond neurons. And I think neuroscience has a lot to tell us about things that are not neurons. For a detailed example of this kind of thinking, I'm going to use morphogenesis, and I'm going to frame it as the behavior of a cellular collective intelligence in anatomical space. Now, in order to have a collective intelligence, you need something binding the pieces towards goals in other problem spaces, and that kind of is exactly the same as it is for neurons in the brain. Bioelectricity, or electrophysiology, is another way of putting it. And I'm going to talk about how this is what I think is the evolutionary precursor of the central nervous system and its functions. I'm going to show you that we now have the tools to read and write memories into this unconventional collective intelligence, and that gives us applications in birth defects, regeneration, and cancer. And towards the end, we'll talk about this idea that there are interesting symmetries across fields, in particular developmental biology and neuroscience, which mean that tools and concepts can be ported in both directions. And I think that's really going to enrich both fields. So the talk comes in three main sections. First, we will talk about some introductions, and really some ideas to broaden the usual kind of focus on conventional animals, conventional brains, and so on. The first thing I would like us all to remember is that we all started life as a single cell. So we all start life as a piece of what people call just physics. I really don't like that term, but a lot of people will look at something like this and they will say, well, it's a little blob of chemistry and physics. This is an unfertilized quiescent oocyte. And then there's this amazing process of embryonic development, which results in one of these complex creatures, or even maybe something like this. And all of us take this journey. All of us go from physics to having a complex metacognitive mind. And it is important to realize that during this process, there is no magic a kind of a bright red line, at which point you go from being a physical chemical system to having goals and memories and preferences and so on. This is something that has to develop slowly and gradually. So I'm interested in continuous views of this process. And in particular, because if we take both developmental biology, evolution, and in fact, bioengineering and augmentation and things like that seriously, we realize that it's really a continuum in many ways, that there is no such thing as a standard pure cognitive system that's such as, for example, a human, which people study. And so that means that we need frameworks for what is now called diverse intelligence. And so I work in this field and I'm trying to develop a framework that will allow us to recognize, create, and relate to truly diverse intelligences. And this means not just primates and birds and things like that, and maybe an octopus or a whale, but also really strange creatures, colonial organisms, swarms, synthetic organisms that have never existed before, artificial intelligences, both embodied and software, and maybe even someday exobiological agents. You know, after all, the Earth is an N of one example of a lineage. Maybe there are very alien kinds of life out there. So what I'm interested in is a kind of framework that allows us to think about all of these kinds of things together. And of course, I'm not the first person to try for this. For example, here is an attempt by Rosenbluth, Wiener, and Bigelow back in the 40s to come up with some general waypoints along a continuum, all the way from passive matter, using kind of a cybernetic kinds of notions, all the way up to the kinds of things that humans enjoy. And so our work in our lab spans from very basic kind of philosophy of mind and things like that, to conceptual frameworks, to software implementations, and bench work to try to test these ideas and develop applications. And so I really like it when these philosophical ideas actually give us new ways to move experimental work forward, and I will show you some of those. So if we think about something I call the axis of persuadability, this is just a kind of continuum all the way from very simple machines all the way through to human and up. What we find is that different kinds of systems on this continuum are amenable to different kinds of interactions. So here, really, it's just rewiring. There's nothing else. You're not going to convince or reward a clock or anything like that. Then you have some systems that are kind of cybernetic sorts of systems where you can reset the set point, and you don't really need to know all the details of how they work, but if you reset the set point, then they will do interesting things. And then we move up to systems that can learn, so various types of reinforcement strategies, inference, and so on. And eventually very, very complex systems that can be greatly motivated by very low energy stimuli that encode reasons to do things. And so there's this whole kind of continuum that is amenable to different tools. So we use different tools down here than we use down here. And that means that we should really try to get it right. For any novel system, we should try to figure out where it lands on this continuum. And a lot of folks have assumptions, sort of philosophical commitments to things, like they will look at some cells and say, there's no way that cells are anywhere over here. Cells have to be simple physical chemical machines and so on. And I really think that it's an empirical question. In other words, it's not something you can have armchair commitments to. You have to make a hypothesis as far as the problem space that these operate in and what kind of tools are going to let you have optimal control. And then you can do experiments, and then we can find out, right? And so there are all sorts of different techniques for doing that. So given that we arise from a single cell through this process of complex development, at least some people say, at least we are a true unified intelligence. We're not really a collective intelligence like ants and beehives and things like that. Maybe those can be called a collective intelligence, but they're not like us. We're a true unified intelligence. And I want to stress the fact that this is really not a good way of thinking about it. You know, for example, René Descartes really liked the pineal gland because there was one of them in the brain, and he felt that the unified human experience should have a single, not a duplicated, but a single kind of point of contact in the brain. But if he had access to good microscopy, he would see that there isn't one of anything. You know, this pineal gland, in fact, is made up of many, many cells. Each one of these cells has all of this stuff inside of it. And so it really becomes clear that we are all collective intelligences. We are all made of parts. There is no such thing, to my knowledge, as a kind of indivisible sort of diamond of intelligence. We all have parts. And this is the sort of thing that we are made of. This is a single cell. And this happens to be a lacrimaria. It's a free-living organism. It has no brain. It has no nervous system. It handles all of its anatomical, metabolic, physiological, and behavioral needs at the one cell level. So here it is, and it's a rather amazing little creature. This is the material that we are made of. We're not made of passive matter. We're made of this kind of agential material that used to be independent organisms and have all sorts of competencies and agendas and so on. Now, one interesting thing that happens is that, for example, when you train a rat for something like when I press a lever, I get a reward. You might ask, who is the owner of this amazing associative memory that it formed? I mean, the cells at the bottom of the feet were the ones that interacted with the lever. The cells in the gut are the ones that received the sugar reward. Who owns this association between this and this? And so here you need to start thinking about the whole animal. It's the rat. It's not, there is no single cell that had both experiences. So no individual cell had both experiences in a way that would allow it to form this association. So now we have to bind these cells in some functional way so that you can have this emergent creature, this rat, which knows things that none of the individual components know. And so this is what we want to talk about. We want to talk about that cognitive glue that enables these subunits to form emergent beings that have memories and preferences and behavioral repertoires in different problem spaces. I want to talk briefly about a couple of unusual examples just to warm up, because many people spend their lives studying, let's say, mammalian brains or something like that. And I want to talk about some unusual scenarios that can help us think in broader directions. So first of all, there's this idea that we have a significant metamorphosis in certain creatures that really helps us to think about how behavior and bodies come together and how they intersect. So this is a caterpillar. A caterpillar is a soft-bodied kind of creature that requires a very specific kind of controller when there are no hard elements on which to push. So running a caterpillar type of body is quite a specific task. And so it has a brain that's well suited for that process. And it eats leaves and it lives in a two-dimensional world. What it has to turn into is this: a butterfly that's a hard-bodied creature, a completely different controller. It lives in a three-dimensional world, flies around and drinks nectar, doesn't care anything about leaves. During this process, the brain is mostly taken apart. Many of the cells are killed. Most of the connections are broken. Really radical reorganization of the brain to get from here to there. But the amazing thing is that if you test butterflies and moths, they still have the memories that you trained the original caterpillar on. And so this work has been done and it's reviewed here. They keep the memories, and not only does memory persist, this remarkable reorganization of the substrate. You know, when I talk about this to computer science audiences, this is a huge challenge. We don't have any substrate for computation that could do this. So not only have the memories persisted, but in an important way, they're generalized, because when you've trained the caterpillar to find leaves on a particular, let's say the particular type of a disc, a colored disc, when the butterfly goes there, it's not because it remembers the leaves. It doesn't care about leaves. It doesn't want leaves. What it's generalized is the concept of food. So both leaves and nectar are now food. So there's this generalization that happens. And all of that happens through a complete reorganization of the brain and the body. It gets even more impressive in animals like this. These are planaria. These are flatworms that regenerate every part of their body, including their brain. And so what McConnell discovered in the 60s, and then we later showed using modern techniques about 10 years ago, is that if you train them to for example, to find food on this particular little substrate, this bumpy substrate, so it's place conditioning, you can remove their heads. The tail end here sits and does nothing. It has no behavior until the brain is formed. So they form their centralized brain, and their head and everything is reformed. And at that point, you can show that they've retained the original information. So here you have information presumably encoded somewhere outside of their brain, imprinted onto the new brain as it develops, so that it can run behavior. And so all of that is discussed here. And that actually brings up all kinds of interesting philosophical questions, in the sense that if you chop them into pieces and these pieces keep the original information, then have you really created multiple individuals which are, in a sense, identical to the original? This is kind of the old malfunctioning transporter experiment from Philosophy of Mind 101. And then I want to show you another example of cognitive plasticity, this time in a vertebrate model. So this is a tadpole. So here's the brain, here are the nostrils, the mouth. And what you'll find is that there are no eyes here. We've prevented the eyes from forming, but what we've done is put an eye on its tail. And I'll show you in a few minutes how we do that. So when you put an eye on its tail, it makes one optic nerve. That optic nerve comes out. It in fact does not go to the brain. Its synapse is sometimes on the spinal cord here, sometimes it actually in the gut, sometimes out here somewhere. But in these cases, the amazing thing is that these animals can see perfectly well. We've created a machine that tests them on visual assays, so visual learning cues, and turns out they perform quite well. There is no evolutionary adaptation needed. So we create these animals. Their sensory motor architecture is completely novel. So instead of having the eyes here where they belong, connected to the brain, there's this weird patch of tissue on their back that is light sensitive and feeds information somewhere into the central bus. And then they can this brain can use that in its behavioral repertoires, no problem. So this plasticity and the ability of the body to change both in a developmental timescale and an evolutionary timescale is something that's really important and we really need to understand. And we think that it is intimately tied to our fundamental multiscale competency architecture. This is the idea that our body from the lowest molecular level up through actually the level of multiple individuals and various swarms, every layer is competent in its own space. In fact, some maybe in multiple spaces. They all solve problems. They're not merely structural nested dolls, but actually all of these things solve interesting problems. And in particular, and we'll get to this towards the end, I really like this quote from Fabrizio Benedetti, who studies the molecular mechanisms of placebo and nocebo effects. He says, words and drugs have the same mechanism of action. The idea that this multi-scale architecture, these levels are not independent. They are functionally linked. And so things that happen at the high cognitive level eventually filter down to the molecular level and vice versa. So there's a constant flow of information up and down. Now, when I say intelligence in various spaces, what I'm referring to is the idea that we as humans are pretty good, actually not great, but pretty good at detecting intelligence in medium-sized objects moving at medium speeds through three-dimensional space. So birds and mammals and things like that. And that's because most of our sense organs face outwards, and we have a very characteristic spatiotemporal scale on which we are able to perceive intelligent behavior. Now, there are other spaces. There's the space of gene expression. There are the space of physiological states. There's anatomical morphospace, which is the space of possible anatomical configurations. And you can think that if we had evolved with a sense organ facing inwards, let's say able to test 20 different parameters or a blood chemistry, we would have evolved with a primary intuitive feeling that we live in a 23-dimensional space, that our liver and our kidneys are intelligent agents navigating that space in a way to solve various problems and so on. But this is very alien thinking to us because we do not perceive any of these spaces directly. And our various body components, in fact, solve problems in these various spaces. So for example, it turns out that even molecular networks, so something as simple as a deterministic gene regulatory network or a chemical pathway, can have 6 different kinds of learning, including Pavlovian conditioning. And so on. So even from the lowest forms of our components of the body, already some of the components of that are tractable to techniques of behavioral science and neuroscience are already there with us. And in fact, one hypothesis that we've been working on is this idea that what evolution basically does is pivot some of the same tricks. And there's this fundamental trick about navigating various spaces, but it pivots this through different spaces, maybe making some specializations, but basically all the way from metabolic spaces at the beginning of life, all the way through to eventually linguistic spaces, and then the need to keep a straight narrative in a complex linguistic space. And of course, this is the traditional behavior is what most people in the field study. Okay, so what we just talked about is this idea that we really need to broaden some of our ideas around what implementations might support different kinds of intelligence, and this plasticity that is underlined by the fact that all of the components are actually solving problems as well. So what I want to do next is talk about this example of a really unconventional agent And this is a collective intelligence that lives in morphospace. And just very briefly, I'll mention evolution, but we could talk for a long time about that. But so this is going to be our example of how do we use techniques from and concepts from so both bench techniques and concepts from neural and behavioral science to understand really a field that is mostly considered to be quite a different field. Now, Alan Turing, of course, needs no introduction, but he was very interested in minds broadly conceived. He was interested in intelligence, in fact in minds, in unconventional substrates and computation, and what is the essential element of being a mind and being able to solve problems and so on. So he specifically was interested in problem-solving machines and intelligence through reprogrammability. Now, it turns out that he also wrote this paper, which is really interesting. He wrote a paper on the chemical basis of morphogenesis, which is a very early model of, it's an early mathematical model of the kinds of processes that might organize a living body from scratch. So this idea of symmetry breaking, emergence of complexity, and regulation of patterns. Now, you might wonder why would somebody who is interested in computer science and fundamental mathematics be looking at models of diffusion and things like that. And I think it's because he saw something very profound, which is that there is a really strong symmetry between these two fields. This idea that the processes by which the body comes to be are really an important mirror of the processes by which minds come to be, and the idea that understanding intelligence and understanding the scaling of cognition in the universe may be quite similar to how we understand where bodies actually come from. And I think that's very profound. And unfortunately, he didn't talk about this directly, as far as I know, in any of his writings. He didn't live to really explore it, but I think he was really onto something. So let's just ask ourselves, where do bodies come from? So this is a cross section through a human torso, quite reliable in most births. And you can see this incredible complexity, this order. Everything is in the right place, the right shape and size relative to the right stuff. And so you might wonder, where is this pattern encoded? Right? Where does this pattern come from? If it's so reliable, where is it? And of course, we all start life here as a bunch of embryonic blastomeres, and eventually you get this. So where is this pattern? And a lot of people will say, well, it's in the DNA, right? It's in the human genome, but we can read genomes now. We know what's in the genome, and what's in the genome is nothing directly related to this. It doesn't specify anything like this. The genome specifies the micro level hardware that every cell gets to have. And so the proteins is what the genome specifies. And so now we need to understand how this works. And so now we know the genome doesn't directly specify this any more than the termite genome specifies directly the shape of the termite mound, or the spider genome specifies the geometry of the web that it's going to build. These are all emergent features from the computations, the physiological computations that hardware performs. And so now we need to understand, how do these cells know what to make? How do they know when to stop? And in particular, though we won't talk about it much in this talk, but what else can these cells build? Like how far can you push them? Could you make them build something completely different? And then regenerative medicine asks, what would you have to do to repair it? In other words, how do you convince cells if something is missing or damaged? How do you convince cells to go back to the normal pattern? We're very interested in this thing, which I call the anatomical compiler. And I thought of this when I was thinking about the end game of our whole field. Like what are we going for, such that when it's done, we can all go home. And the idea is that at some point in the future, you'll be able to sit down in front of a computer system and draw the plant or animal that you want, or the organ, or the weird biological robot. It doesn't matter. You should be able to draw some biological structure. And if we knew what we were doing, the compiler would convert that anatomical description into a set of stimuli that would have to be given to cells to cause them to build that particular structure. Now, why do we need this? Well, first of all, most biomedical problems would go away. So everything related to birth defects, traumatic injury, cancer, aging, degenerative diseases, all of that would go away if we had the ability to tell a collection of cells what to build, because then you could build and rebuild organs and other structures at will. Now, the important thing about this is that this is not meant to be a 3D printer. The point isn't to micromanage and put cells where you want them to be. It's really more of a communications device. It's really a translator between your anatomical goals and the set points that guide cell behavior. Now, why don't we have this? Why are we so far away from this? I mean, molecular biology and genetics have been cranking really hard for decades now. Why don't we have this? I just want to point out a very simple example. So this is a baby axolotl. Axolotls have little forelegs. At this stage, here's a frog larva. They do not have legs at this stage. We make something in my lab called a frogolotl. So this is just a chimera of an axolotl and a xenopus embryo. And both of these are sequenced. So you've got the axolotl genome, you've got the frog genome. Now I ask a simple question: are frogolotls going to have legs or not? And the fact is, we have no idea. No, we can, of course, we can look empirically, but nobody can make a prediction. There are no models, despite the availability of genetic information. There are no models that will make a prediction on an experiment like that. In fact, the genomic data can't even be used to guess what the actual shape of the organism is going to be, unless you cheat by comparing that genome to something else that you already know what it is. So here's where we are today. The field is very good at manipulating cells and molecules, so we can get information like this. We're really quite a long way away from large-scale form and function control. And I'm going to argue that that is because we are currently where computer science was in the 40s and 50s. This is what programming looked like in the 40s and 50s. You literally had to interact with the hardware. You had to physically rewire it. But the reason that nowadays you don't get out your soldering iron and start rewiring your laptop when you want to go from Microsoft Word to PowerPoint is because we have figured out that there's a ton of power in high-level interventions that reprogram and take advantage of a certain kind of hardware. And I'm going to argue that biology is exactly that kind of hardware, which again, for neuroscience, this is not exactly news. But for molecular and developmental biology, this is a different way of thinking. And this idea that we've only begun to scratch the surface of what's actually possible. We have not really understood the amount and the type and the tractability of information processing and intelligence or problem solving at different levels of biology. And what do I mean by intelligence? Well, I like William James's definition. William James said that intelligence is the ability to reach the same goal by different means. So it's a cybernetic definition. It doesn't talk about brains or whether you were engineered or evolved or anything like that. It talks about the difference between two magnets trying to get together, which if faced with a barrier will simply stand there pressed up against the barrier. They're unable to walk around, temporarily get further from their goal in order to finally reach it. They have no delayed gratification ability. Very simple system. Romeo and Juliet, on the other hand, and this is James's example, have all kinds of complex abilities, including planning and everything else to try to get their goals met. In between, you have all sorts of things, cells and tissues and self-driving vehicles and animals of different types. And so this ability to reach the same goal despite novelty, despite novel scenarios and so on, is a kind of measure of this intelligence. And it's important because these competencies, these capabilities, can be taken advantage of in therapeutic contexts if we understood how to deal with them. So what kind of collective intelligences do cellular swarms have? I'm going to show you a few examples. One is that while development is very reliable, so almost always you start with a human egg and you end up with a human organism. Actually, it's not hardwired, because what you can do is take early embryos, cut them in half, or in fact in many pieces, and you don't get half bodies, you get perfectly normal monozygotic twins. And so there's this ability to navigate to the ensemble of goal states corresponding to some kind of a normal human target morphology from different states. And so, of course, I'm collapsing a very complex anatomical morphospace onto just 2 dimensions to show how they all navigate to that set point, avoiding various local maxima along the way and so on. That kind of regulative development is not just for embryogenesis. It also works in some animals. It works during their whole lifespan. So this is a salamander. So they regenerate their limbs, their eyes, their jaws, their ovaries, their spinal cords, and so on. And what's interesting about this process is that if you cut it anywhere along, for example, this limb, they grow exactly what's needed, no more, no less, and then they stop. That's the most amazing thing about regeneration, is that it knows when to stop. When does it stop? Well, it stops when a correct salamander arm has formed. And so this is a kind of anatomical homeostasis. You can start off at any configuration and it will keep going in the right way until you get to the right thing. By the way, humans can do this a little bit. This isn't just for worms and salamanders. Humans regenerate their liver. Even the ancient Greeks knew that. I don't know how they knew that, but they did. Human children will regenerate their fingertips up to a certain age, and deer regenerate huge amounts of bone, vasculature, innervation, and skin every year. So up to 1.5 centimeters of new bone growth per year, per day, when they're growing back their racks. And I don't have time to talk a lot about this, but this idea that what evolution is working with is a kind of a competent material where cells will really correct for all kinds of defects and so on, results in a very interesting intelligence ratchet, because that competency makes it hard for selection to see whether the fitness of the animal comes from the quality of the genome or from the fact that the actually structural genome wasn't great, but the competency made up for it. And the more that happens, the more effort from evolution actually goes into ratcheting the competency mechanism. So it's a very interesting thing. We can maybe talk about that afterwards. So now that we've seen that collectives of cells can solve various problems. Remember, the problem that they solve is how to walk a particular path through anatomical morphospace that starts out as a single cell, for example, or a healthy organ in the case of regeneration, and then is deviated from that to a different location, and then knows how to come back despite various perturbations. And there are actually remarkable examples of this that I don't have time to go through here, but they're all discussed in our papers. So now that we see that ability to navigate morphospace is the thing we want to explain. Now there's an obvious thing we want to look for, which is what is the cognitive glue? How are the cells all bound together to the same construction project? No individual cell knows what a finger is or how many fingers you're supposed to have, but the collective clearly does. And so we need to understand how the individual cells are bound. And so we're going to talk about bioelectricity. Now, just to remind you of a simple experiment that forces that kind of question is this. This is something that we discovered a few years ago. So here's a tadpole, two normal eyes at this point, brain, nostrils, mouth right here. These tadpoles have to become frogs. In order to become frogs, they have to rearrange their face. So everything has to move. The eyes have to move, the jaws, the nostrils, everything has to move around. And it was thought that this is a very hardwired, meaning low intelligence process, because all tadpoles look the same, all frogs look the same. So as long as you somehow encode specific movements, in other words, direction and amount for each of these organs, you will end up from a normal tadpole to a normal frog. So we decided to test that. And the way you test the competency of these kinds of systems is by perturbative experiments. You cannot do it just by observation. You cannot do it by only modeling. You have to perturb them by, as William James said, putting various obstacles in their way and seeing what they do to respond. And so what we did was we created so-called Picasso tadpoles. Everything is in the wrong place. The eyes on the back of the head, the mouth is off to the side. I mean, the whole thing is scrambled. And what we found out is that these animals actually end up being pretty normal frogs, because all of these organs do not just go in the same path that they do in normal animals. They will go in novel, abnormal paths. In fact, sometimes they go too far and they have to double back until they get to the correct frog face, and then they stop. So what the genetics actually gives you is an error minimization scheme, a system that can recognize that it's in the wrong state and take corrective action to get where it needs to go, to complete that journey in morphospace, despite being started in a very abnormal configuration. And again, this configuration is at the large scale. So it's not that the individual eye cells are sprinkled throughout the whole head. It's the eye as a whole has to move, and the mouth and everything else. This is truly a collective decision-making problem. But it raises a fundamental question. How does it know what the correct pattern is? What is the set point? How does it know what the frog face is supposed to look like? And so to try to formalize it a little bit, you can think of it as, first of all, this is the standard kind of story that you're told in developmental biology textbooks, which is that there are gene regulatory networks. They make proteins, they interact with each other in various ways. There's this process of emergence, and it's a very complex process, because lots of actually simple local rules are being carried out again and again, and out comes something complex like this. So this is a feed-forward, open-loop kind of process. And this certainly does happen, and there are many simple processes that give rise to complexity. I mean, this is true, but this is only part of the story, and I don't think even the most important part, because what happens here is something very interesting. If you deviate from this state by injury, mutation, teratogens, many other ways, what happens is that activity kicks in both at the level of physics and at the level of genetics, that actually tries to reduce the error and get you back where you need to go. So this is not really an open loop process. This is a process of anatomical homeostasis. And on the one hand, this is maybe not so surprising. I mean, of course, biologists know all about homeostatic loops. On the other hand, there's a couple of weird things here. One is that typical homeostatic loops have a scalar as a set point, a single number. So maybe hunger or level or pH or something like that. In this case, the set point is basically a complex anatomical structure, not to the individual cell level or anything like that, but really a fairly complex data structure here. That's first. And second, you know, certainly in behavioral science, you can talk about at least some kinds of animals having goals and doing things for reasons, but typically in cell and molecular biology, we are told to avoid that. You're not supposed to think about systems having any kind of a future goal, or you know, you're supposed to sort of feed forward from chemistry and see what happens. So this is kind of a weird way of thinking about it, but it makes a very strong prediction. The prediction is that that as a good homeostatic system, you ought to be able to make changes by rewriting the set point. In other words, you should be able to find the biophysical mechanism of the set point. How does the system know what the correct pattern is? You should be able to decode it. You should be able to rewrite it. And those three things are important in applications, because currently the only game in town is down here. If you want to make changes up here, we are told that, well, you'll somehow have to work out what changes in the lowest level rules are going to give you system level changes that you want. And this process is fundamentally irreversible. There is no, it's a really terrible inverse problem. There's no real way to reverse it back. So that limits the applicability of things like CRISPR and genomic editing, because it's really, with the exception of some low-hanging fruit, it's really completely unclear what genes you would edit to make large-scale changes here. So could we actually find the set point? And this is where we've been looking. Now, in thinking about where to find the set point, we said, well, what's the one example, the one non-controversial example of a biological system that really encodes goals and the pursuit of goals? And that, of course, occurs in neuroscience. So we know what the hardware is. The hardware are these networks of cells that achieve a certain voltage potential by means of ions moving through the ion channels. They may or may not communicate that voltage to their neighbors through these electrical synapses known as gap junctions. And so that's the hardware. The software here, that's mediated by this, it can be read out. So this group made this amazing video of a living zebrafish brain, and you can see all the electrical signaling. And it's the commitment of a lot of neuroscience that this is really the encoding of the cognitive content of this individual. So there's this process of neural decoding that if we were to record all the electrical activity, and if we knew how to decode it, you would be able to read the animal's memories, preferences, goals, potential behaviors, and so on. Right? So that's the process of neural decoding. This physiology is where we think all of that is sitting. The amazing thing is that actually every cell in your body does this. So every cell in your body has ion channels. Most cells have gap junctions to their neighbors. And from the earliest time of development, your tissues are an electrical network that is processing information. Now, we can then try the same kind of decoding research program. Could we decode the bioelectrical signals in non-neural tissues and ask, what are they thinking about? What problems are they solving? And in particular, and so this is much like here, we're tracking the voltage with a voltage reporter. This is an early frog embryo, again, time lapse, of course, again, reporting the voltage so we can see all the electrical conversations that these cells are having with each other as they sort out who's going to be head, tail, and so on. And so there's this basic kind of diagram where what the bioelectric networks in your nervous system are doing is controlling your muscles to move your body through three-dimensional space. Okay, and this can be decoded and modeled and so on. And my argument is that this amazing trick is really an elaboration of a much earlier, evolutionarily, a much earlier system in which all of the cells were running an electrical network to move the configuration of your body through morphospace. And what evolution has done here is a couple of things. One is greatly speed optimize the process. So we're talking milliseconds in neuroscience as opposed to minutes or hours in developmental bioelectricity. And it pivoted space for time, where this is mostly about the control of space. One can argue that nervous systems are mostly about the control of behavior as a function of time. Okay, so there is this, and by the way, evolution caught on to this system as early as bacterial biofilms. This is incredibly ancient and long predates nerves and muscles and things like that. So we then went on to try to develop some of the first tools, the first molecular tools, to read and write this cognitive information out of non-neural cells. Now, no surprise, we took most things from neuroscience because, as it turns out, the tools cannot tell the difference. We try to maintain that difference through different journals and different funding bodies and different educational materials and courses and departments. But actually, all of the tools, both the bench tools and the concepts, poured over very nicely. So it's interesting. Some of that distinction might actually be quite artificial. So we developed some tools to non-invasively read the bioelectric pattern. We also do a lot of computational modeling to try to understand how these bioelectrical patterns result from the activities of channels and pumps and gap junctions and so on. So let me show you a couple of such patterns. This is an early frog embryo putting its face together. And so you can see, again, this is a voltage sensitive fluorescent dye in time lapse. This is one frame out of that video. We call it the electric face. And you can see here that in that frame, you can already see, so here's the animal's right eye, here's the mouth, here are the placodes. The major structures of the face are already laid out in this subtle bioelectrical pre-pattern before the genes come on to regionalize that face and before the anatomy actually starts to form. So as it turns out, this is absolutely an essential pattern memory that is required for forming a normal face. If you move this around, as I'll show you in a minute, the genes and the anatomy will faithfully follow. So that's an endogenous pattern that you might use for monitoring defects. Now, here's and we'll come back to cancer a little bit at the end, but here's a pathological pattern. When you inject a human oncogene, eventually you'll get a tumor. Eventually, it'll start to metastasize like this. But before that, you can already tell where it's going to be. And in fact, you can tell where the regions are that are going to go off of the plan here and revert back to an amoeba-like environment where they're unplugged electrically from the rest of the network. They're not receiving the signals about how to build various organs. They're just amoebas. As far as they're concerned, the rest of the body is external environment. Okay, so these are just some examples. And I show you this, by the way, because this is the easiest pattern to decode. We have many other patterns that are actually quite hard, but this is pretty obvious what you're looking at. So in addition to the imaging, of course, the most important thing is functional, right? So can we manipulate these bioelectrical patterns and show that they're functionally critical for what the morphogenetic collective does next? And here I have to stress, we do not use any applied fields. There are no magnets, there are no frequencies, no electromagnetic waves, no electrodes, nothing like that. We use all the tools of molecular neuroscience and physiology. So we use pharmacology to open and close channels and gap junctions. We can mutate the channels to change their properties in a known way. We can use optogenetics and so on. And so the idea is to take any kind of a region in the animal and use these tools to really just alter the topology and the activity of the bioelectrical network in a very specific way to try to change its decision making as it navigates the anatomical morphospace, right? So whereas the neural decoding was the part with the dyes that I just showed you a minute ago, this is equivalent to the inception of false memories, for example, and people have done this in mice and so on. So I want to show you what happens when you actually do this. So the first thing that happens is remember that little eye spot that I showed you in the bioelectric face. Well, you might ask, what happens if you introduce that same kind of spot somewhere else? You take a memory of eye formation and you falsely reproduce it in other regions of the body that are actually supposed to be doing something else. Well, if we inject specific ion channels that can induce that same type of eye spot that I just showed you a minute ago, In fact, and so you introduce it into cells that are precursors of things like the gut, those cells will happily build an eye. And these eyes can contain all of the right layers, so lens, optic nerve, retina, all that stuff. So they all have them. So this tells you a couple of different things. First of all, it tells you that the bioelectric pattern is actually instructive. This is not about messing up the pattern and making monsters. This is about showing that those patterns can cause specific organ level changes in morphogenesis, right? So not individual cells. We're not telling just individual cells to differentiate a different way. We're telling the whole region to make an eye. So this is extremely modular. We don't have to provide all the information about how to make an eye. We don't know how to make an eye. This is very high level subroutine call, and this only works with high level competent systems, because in very simple machines, you have to be in charge of every aspect of the functionality. You have to program every aspect of what it does. Here, we don't need to do that because the system has built-in competencies. In fact, now the interesting thing about these competencies is that when you're analyzing a system, any system, to make an assessment over what it can do, you're really taking an IQ test yourself, because it is not obvious. And if you don't prompt it in the right way, you don't know. So for example, the textbook will tell you that only the anterior neuroectoderm up here is competent to make eye. And the reason they say that is because the classic experiments were done with what they call a master eye gene, PAX6. And it's true, if you misexpress PAX6, only these cells are competent to make eye. But that isn't the actual best master regulator. And if you do it with a voltage state, in fact, you find out that the competency is much more than you thought it was. Every cell in the body is able to participate in this, including endoderm. Endoderm's not supposed to make eyes, right? So it's important to actually be humble about when the limitations that we see are really a result of our own lack of knowledge about what really is a meaningful signal to the surrounding tissues. And the final thing I want to just point out is that this is a lens sitting in the tail of a tadpole somewhere, an extra lens. The blue cells are the lineage label of the cells that we actually injected with our channel. All of this other stuff here was never manipulated by us. Why does it participate? Well, that's because these cells can do what many collective intelligences do, like ants and termites. When there's not enough of them to do the job, they recruit their neighbors. So there's literally a secondary instruction event that's taking place where these cells tell these other cells, hey, you need to work with us to make this eye. We don't have to program that in. Both the eye induction and the scaling to the task, all of that is a native competency of these collectives if you know how to activate them. And so we can make many things. We can make ectopic forebrain and ectopic hearts and ectopic otocysts and extra legs. So this is actually an optogenetic frog that has an extra leg growing out of its mouth. We can make fins. That's kind of weird. Tadpoles aren't supposed to have fins. That's more of a fish thing, but we'll get to that. So what you can see is that it is possible to rewrite these bioelectric pattern memories in ways that make the collective navigate to different regions of that anatomical morphospace. And we're applying this in our regenerative medicine program. So frogs normally, unlike salamanders, do not regenerate their limbs. 45 days later, there's nothing. But we came up with a cocktail that immediately, within about 24 to 48 hours, triggers pro-regenerative genes like this MSX1 in this blastema. And by 45 days, they've got toes, they've got a toenail, and eventually a pretty respectable frog leg that's both touch sensitive and motile. And the thing about this is that this is not micromanagement. We're not 3D printing anything. We're not controlling stem cells. We're not interacting with this thing at all. 24 hours of stimulation, and then for a year and a half, this leg will actually grow. And so it's the decision-making of the collective very early on. It's not trying to micromanage it. And here, of course, I have to do a disclosure because Dave Kaplan and I have a company called Morphoceuticals Inc., where we're trying to apply the same technology. So a wearable bioreactor and then a payload of modulators that convince the cells that they need to rebuild. And we're now moving into rodents. The next story I want to tell you has to do with planaria, because I really want to hammer this idea of bioelectrical pattern memories being literal representations of navigation in this anatomical space. So these planaria have this amazing capacity that you can cut them into pieces. I think 276 is the record, something like that. And every piece makes a perfect little worm. So we asked a very specific question. Once you've cut this piece like this, how does it know how many heads it's supposed to have, right? And so And so what we found out is that, in fact, there's an electrical circuit that sets a voltage gradient in these pieces that determines how many heads it's supposed to have. And so if you take a normal one-headed worm and you have this body, this worm has anterior genes in the head, not in the tail, like it should, and it very reliably makes this one-headed worm. But that's because it has this bioelectrical pre-pattern that says one head. And so what we were able to do is take an animal, give it a different bioelectrical pre-pattern. This is kind of a mess. The technology is still being worked out, but you can see here now, it's very clear. There's now these two regions. And this animal still has its anterior genes in the head, not in the tail. But if you cut him, he gives you a two-headed worm. And this is not Photoshop, this is real. And something important to stress here is that this bioelectrical pattern is not a map of this two-headed animal. This is a readout of this one-headed animal, which is anatomically and transcriptionally normal, and it will be that way, will stay normally one-headed until you injure him. So this is a readout of a kind of counterfactual memory. It's what I will do if I get injured. It's what does a correct planarian look like, aka the set point of that anatomical homeostatic cycle. So I promised you at the very beginning that if we are to understand how these systems solve problems in anatomical space, we have to know how do they remember what is the correct pattern. And what you're looking at is one simple example of how they encode goal state information. This is literally a kind of a precursor to, I think this is a precursor to this amazing ability of human brains and other animals to time travel, to think about things that are not true right now, things that used to be true or that will be true in the future, things that are not true right now. So that's it. This bioelectrical state does not describe the anatomy at this moment. It describes a goal state that will be pursued if the animal is injured. Now, I keep calling it a memory. Why? Because if you take these two-headed animals, you can cut them again in plain water. So you cut off the ectopic primary head, you cut off the secondary head, and with no more perturbation, they will continue to generate two-headed animals in perpetuity. Now, remember, the genome is normal. There's no genomic change here. This is a very sort of counterintuitive result because it was thought that if you cut off the extra head, this genetically normal fragment, well, of course, it's going to regenerate the normal thing, what the genome says. And so this is why, two-headed worms were first seen in 1903. As far as I know, nobody cut them until we did it in 2008. Because it was too obvious what would happen. I mean, of course, they'll go back to normal. And in fact, that's not what happens. And so what you see here is that the number of heads is not directly nailed down by the genome. The genome gives you a machine that by default generates a memory of one head, but you can rewrite it. It's reprogrammable. Without, in fact, editing the genome, you can physiologically give them a new memory and they will continue to be two-headed. It's long-term stable, it's rewritable, and it has conditional recall. Which is I showed you a minute ago. And so this is, you can see here this video of these guys hanging around. So what we are doing now is we are trying to put together different ways of thinking about systems that have different attractors. So the bioelectrical, the state space of the bioelectrical circuit that corresponds to one head, no head, two head, and so on. And lots of thought in connectionist AI and neuroscience about how electrical networks can store memories of patterns and regenerate those patterns upon damaged or partial inputs. And there's a lot of work to be done to integrate these, but we think they're both different views on the same set of processes. Amazingly, it's not just the number of heads, it's also the shape of the head. So this triangular shaped planarian can be, by manipulating these bioelectrical patterns, could be made to make a flat head like a P. felina or a round head like an S. mediterranea. Again, no genomic change, about 100 to 150 million years evolutionary distance between this and this. It also changes correctly the shape of their brain and the distribution of stem cells like these other species. So what they're doing is this same hardware is perfectly able to visit the attractors in morphospace belonging to these other species. That's normally where these other species go, but this hardware can go there too if properly guided in morphospace. In fact, you can go way beyond things that even look remotely like planaria, and we've seen planaria make things like this spiky form, these kind of cylindrical shapes, and this kind of like combination hybrid shape. And so I think this is what we're looking at here, is what Darcy Thompson in 1917 saw as a latent morphospace around each animal and plant form. And, he drew these axes, and it was entirely unclear what these axes were, but I actually think he's representing the internal encoded map of the collective intelligence of these cells, and that we now have the ability to actually stretch and deform that internal representation. So what we're trying to do is basically the same thing that multiscale neuroscience wants to do, which is to go all the way from the molecular level where different channels and pumps are expressed, to the different bioelectrical, the spatial bioelectrical patterns, to the organ level and a whole animal level type of anatomical order, and ultimately to algorithmic models that describe this behavior, so that we can actually rationally understand and manipulate what's going on. So a kind of a full stack approach here. And I want to show you one example where we've been able to do that, which is this in the frog brain. So here's the normal frog brain: forebrain, midbrain, hindbrain. There are a variety of teratogens, both chemical and genetic, that screw up the brain. And we decided to check whether we could use a computational approach to try to repair this very complex structure. So we made a computational model of the bioelectrics around the pre-pattern that tells this brain the shape and the size that it should be, having characterized this pre-pattern years before that. What we were able to do is to create a model that, in the case of, and I want to show you kind of the most extreme case. So we've done nicotine and alcohol and different things, but here's the most extreme case. I think most of you will know the Notch gene, which is a very important neurogenesis gene. And when Notch is mutated to a dominant active form, what you see is that there is no forebrain. The midbrain and hindbrain basically are a big bubble. These animals have no behavior, profoundly disabled. And what we did was we created a computational model of this, and we asked the model, what channel could we open or close to get back to the correct bioelectrical state? And so it focused in on this. It's a very interesting channel with some specific properties, this thing called HCN2. And we found that when we upregulated HCN2, either by injecting RNA encoding it or with HCN2 openers, you basically get back to a normal brain. Normal, anatomically normal in terms of gene expression, and they get their IQs back. So indistinguishable learning rates from control animals. All of this while still having that Notch mutation. So at least some types of genetic defects, meaning hardware defects, can be fixed in software. I'm not saying they all will be like that. There will be many that are not like that. But it really is amazing how much control you can have at the software level and override what really is a pretty profound hardware defect. And so what we're doing now is we're trying to create this platform, and you can play with an early version of it here online, that ultimately is going to have different tissues and organs, both normal and transformed. And you can go on and say, okay, I have an abnormal bioelectric pattern in some region, and this thing will computationally model it, and then it will give you a list of channels and pumps that are expressed in that region, but which we know from scraping transcriptomics profiles. And it will give you targets to try to get the incorrect pattern into the correct pattern. And of course, then you can pick drugs, because something like 20% of all drugs are ion channel drugs. So this is a huge toolkit of potential electroceuticals if we understood the modeling. But the idea again is not to micromanage the state of any one cell. The idea is to communicate with the collective and try to get it to move to different regions of that morphospace and exploit the different competencies that it has for repair, for scaling of activity, and so on. And so kind of now pulling back, so I'm almost done here. And so pulling back, all the details, by the way, are in these papers. I want to put forward a view of biomedical interventions as fundamentally in two categories. There are all the bottom-up ones that seek to really interact with the hardware and micromanage the symptoms and the functionality. And then there are all of this stuff, which is really just beginning to be exploited. And the idea is that top-down, you can actually take advantage of the many competencies of your material. And so there's something very special here for this, because neuroscience brings something extremely important. It's a unique, it's really a unique field for all of this. And we've been doing this applications in birth defects and normalizing cancer and regeneration and so on, because because in neuroscience, it's basically the only field where you can see how electrophysiology mediates from reasons, so from high-level cognitive intent to molecular causes. And what I mean is just simply that, you know, if I told you that with the power of my mind alone, I could depolarize 30% of my body cells, you might think that's crazy, or you might think that that's some special mind-body medicine kind of thing, maybe some biofeedback or something. But of course, it's absolutely normal 24/7 event that happens during voluntary movement. So in the morning, when you make this high-level executive decision that you're going to go to work, that information flow has to trickle down to the opening and closing of ion channels in your muscle cells to get them to fire so that you can get out of bed and go to work. That incredible architecture that our bodies have, that link all the way from high-level cognition through all the circuits that you all study and everything else, and down into the molecular control of cells, is a unique multi-scale understanding that really is still missing in other fields and can really be imported to great use. So my gut feeling is that all of this means that future medicine in these areas, so regenerative medicine, is going to look more like a kind of somatic psychiatry, where you're literally communicating with the collective and manipulating its prior memories, its measurements, its beliefs about the landscape, the energy landscape that it's in these different spaces, and so on. And we have a lot to learn from how multiscale neuroscience does this. So what I've told you today is that intelligence and navigation of problem spaces is, I think, a kind of generic concept that's applicable far outside of standard behavioral science. We're all collective intelligences, and that's why I think that neuroscience and the deep insights and the fact that its tools really can be broadly applicable, and its concepts, you know, we use everything from active inference and perceptual control theory and but you know, bistability and all this kind of stuff is multi-scale processing in the right, and all of these things we use outside of the nervous system to help us achieve better control in regenerative medicine. And I've talked about the behavior of a collective intelligence of cells in morphospace as underlying these amazing properties of development, of regeneration, and so on. And I really think that this is where the brain learned its amazing tricks. And so the last thing I just want to show you very briefly is this idea that there's this really interesting invariance across fields that can enrich both, right, developmental biology and neuroscience. And for many years, I was saying to my students that, you know, you can take almost any neuroscience paper and just do a find replace. And every time it says neuron, you just say cell. And every time it says millisecond, you say minute or hour. And then you've got yourself a nice potential developmental biology paper full of hypotheses to explore. And so we finally, with two collaborators, Thomas O'Brien and Joel Stremmel, we actually made an AI tool, and you can all play with it here, that does this in a very smart way by mapping a few terms to each other. And you can take any abstract in a neuroscience paper and see what the corresponding developmental biology paper looks like. So you can see here, this is a beautiful paper by Panotsis and Miller. And so you go on here and you feed this abstract, and what it gives you here is a very plausible set of claims that is testable in developmental biology. And so we are developing these kinds of tools to help exploit the invariances between fields to really enrich both sides of this equation. And the final thing I just want to show you is that I think that because of this amazing interoperability of life and the fact that cells and tissues are a kind of material that are happy to work with all sorts of novel configurations and new materials and so on, all of existing biology, everything that Darwin meant when he said endless forms most beautiful, is like this tiny corner of option space. And the future, meaning the next couple of decades, we're going to be surrounded by all of these things that already exist, especially in neuroscience. So cyborgs, hybrids, different kinds of chimeras and combinations, all kinds of bodies and minds that we've never seen before, that have never been on the tree of life with us. And we are really going to be looking for insights from diverse intelligence research to expand the way that neuroscience and behavioral science thinks about agency, cognition, memory, and how we are going to ethically relate to these creatures that actually don't look anything like us, and you can't use the familiar kinds of things we use when talking about brain organoids in a dish or something. What does the brain look like? Is it like a human? It doesn't need to be like a human. There are going to be all sorts of really wild combinations that we need to think about. So all of this is discussed in these various papers, and I just want to thank the postdocs and the students that did all the work. Our many collaborators and all of our technical support. Again, here are 3 companies that support our work, and many other funders here, and most of all the model systems, because the animals do all the heavy lifting. So thank you so much, and I'm happy to discuss.